Good evening, ma'am.
Good evening, sir. You can start now, sir.
Welcome back. The MCQ is based on the chapter Biomolecules, and we'll be finishing the 12th syllabus possibly today. And these are direct questions, not of much of reactions involved. So we'll rush through this and finish up all the MCQs possibly today. Now, the first question: which of the following is the sweetest to sugar? Sucrose, glucose, fructose, and maltose. We have learned monosaccharides and disaccharides are sweet to taste and soluble in water. Polysaccharides are not sweet to taste, and they are insoluble in water. Now, example for a polysaccharide: starch, cellulose, glycogen. All these are examples for a polysaccharide. Now, all these here, what they have mentioned, all are sugars. All are sugars. The sweetest among all these sugar is fructose. Fructose is also called as fruit sugar. So fructose, in honey, fructose is present. So the sweetest sugar amongst all these is fructose. The next question: alpha D plus glucose and beta D plus glucose are anomers, epimers, enantiomers, geometrical isomers. Now, if you remember, we learned the structure of glucose like this. The 6-carbon atom. And also we learned the glucose structure like this, the alpha and beta structure. I'm not writing the complete structure. Now, if you can identify here, the hydrogen atom of the fifth OH group of the 5th carbon atom will attack oxygen. Therefore, this pi bond breaks. When this pi bond breaks, the 1st carbon atom and this hydrogen attacks here, the 1st carbon atom and the 5th carbon atom is bonded through oxygen. You can see the OH group is on the right-hand side. Similarly, here also, the hydrogen atom of the OH group of the 5th carbon atom attacks the hydrogen atom. When it attacks the hydrogen atom, the pi bond breaks. Now, the 1st carbon atom and the 5th carbon atom are bonded through oxygen. Now, you can see in the first position, the configuration of carbon is different. In this structure, the OH is on the right-hand side. Here, the OH is on the left-hand side. All the other structure, the part of the structure is same. So, when the configuration of the 1st carbon atom is different, then we call those kind of isomers as anomers. So, alpha D plus glucose and beta D plus glucose are anomers. The third question: coupling between base units of DNA is true. Coupling between the base units of DNA. Coupling is bonding between the base units of DNA is true. Hydrogen bonding, electrostatic bonding, covalent bonding, van der Waals force. So the bonding between the bases in DNA is through hydrogen bonding. So the double helical structure of DNA. So the helix are held together by hydrogen bonding. The segment of DNA which acts as an instrumental manual for synthesis of protein is nucleotide, ribose, gene, nucleoside. The segment of DNA which acts as instrumental manual for the synthesis of protein is a gene. This is totally a biological concept. Now, fifth question, which of the following structure represents a peptide chain? Now, we clearly learned what is a peptide bond. C double bond O, NH bond. This is called as a peptide bond. CONH bond. Otherwise, we can write CONH bond will be called as a peptide bond. Now, which of the following structures represent a peptide bond? So you have to look into all the structures. Now, CONH we have here, CONH we have here. Here we do not, this we have CONH here. And here it is not a peptide linkage. Here you have CONH. And here this is wrong. You can see it. CO2NH groups are there. Therefore, this is also wrong. Here you can see CONH, CONH, CONH. Here NH is there, no CO, close to each other. This is also wrong. Therefore, the answer is option C. Option C represents a peptide chain. You can see here one CONH bond, two CONH bond, and also we have three CONH bond. Hence, the answer for this question is option 3. On hydrolysis of starch, we finally get hydrolysis of starch. So starch, when we hydrolyze in the presence of an acid or an enzyme diastase, we get maltase, and this maltase further on hydrolysis, finally we get glucose. So starch on hydrolysis, finally we get glucose. This is maltose. And finally we get glucose. The seven from each other due to the difference in carbon atom with respect to its conformation, configuration, number of OH groups, size of the hemiacetal ring. It differs in the configuration. Arrangement of OH group in alpha, it is on the right hand side. OH is on the right hand side. In beta, the OH is on the left hand side. So it is a configuration, it is not conformation. The next question. Glycolysis is oxidation of glucose to pyruvate, conversion of glucose to heme, oxidation of glucose to glutamate, conversion of pyruvate to citrate. Glycolysis is the process in which glucose is broken down to produce energy. Produce energy. A process in which glucose is broken down to produce energy is called as glycolysis. It produces two molecules of pyruvate, ATP, adenosine triphosphate, NADH, and water. So therefore, the answer for this question is option 1, oxidation of glucose to pyruvate. Glycolysis is a process in which glucose is broken down to produce energy. It produces two molecules of pyruvate, ATP molecule, NADH, and water. Sucrose, when dissolved, when sucrose in water is dextrorotatory, plus 64.4 degrees. When boiled with diluted HCl, the solution becomes levorotatory, minus 39.9 degrees. In this process, sucrose breaks into. Now we know that sucrose on hydrolysis, sucrose is a disaccharide. This on hydrolysis, in the presence of an acid, we get D plus glucose. One is glucose unit, the other one is fructose unit, D minus fructose. Hence, the answer for this question is option 3, D glucose and D fructose. Sucrose is a disaccharide. The next question, which of the following is the correct statement? Starch is a polymer of alpha glucose. Amylose is a component of cellulose. Amylose is also a component of starch. Proteins are composed only of one type of amino acid. It has many different types of amino acids. In cyclic structure of fructose, there are 6 carbon atoms. There are 5 carbon atoms. So starch is a polymer of alpha glucose. This is the right statement. So answers option 1. 11th question. Glucose molecule reacts with X number of molecules of phenylhydrazine to produce osazone. To produce osazone. The value of X is. The value of X is. Glucose reacts with phenylhydrazine to give glucose phenylhydrazine, which is soluble in water. Which is soluble in water. So glucose reacts with three molecules of phenylhydrazine, which is soluble in water. Which is soluble in water. Therefore, the answer is option C. It is glucose molecule reacts with three molecules of phenylhydrazine to form osazole. Chemically considering digestion is basically anabolism, hydrogenation, hydrolysis, dehydrogenation. Chemically considering digestion is basically hydrolysis. The next question. Among the following non-reducing sugar, glucose, fructose, maltose, sucrose, and galactose. So non-reducing sugar which does not reduce. Sugar which does not reduce. Tolerance reagent and Fehling's solution is called as a non-reducing sugar. So non-reducing sugar example is sucrose. If a sugar should be a reducing sugar, it should have free aldehyde or ketone group. So in case of sucrose, no free aldehyde group or ketone group is there. Therefore, sucrose is an example for non-reducing sugar. Very important question. So sucrose is an example for non-reducing sugar. All other monosaccharides are reducing sugars. Now, the next question, the two forms of glucopyranose obtained from D-glucose is called as isomers, anomers, epimers, enantiomers. We learned this question. Alpha D plus glucopyranose and beta D plus glucopyranose. The structure only differs at the first carbon atom. The OH configuration differ only at the first carbon atom. For alpha, the OH is on the right-hand side, and for beta, the OH is on the left-hand side. Therefore, they are called as anomers. They are called as anomers. Hence, the answer is option 2. The next question. Sucrose on treatment with concentrated HCl produces glucose and fructose and maltose. It's a direct question. So sucrose when treated with concentrated hydrochloric acid, this will undergo hydrolysis. Sucrose will undergo hydrolysis in the presence of a strong acid like HCl to form one unit of glucose and the other unit of fructose. Hence, the answer is option 3. The next question. Mixture of glucose and water can be separated by distillation, crystallization, sublimation, all the above. The mixture of glucose and water can be separated by distillation. Option 1. The next question. Glucose and mannose. Glucose and mannose are C1 epimers, anomers, C2 epimers, C4 epimers. Now we will write the structure of glucose. This is the structure of glucose. This is H, this is OH. And mannose, we will write the structure. If you observe what I've written, you can see the configuration is different only at the second carbon atom. In the second carbon atom, the OH is on the right-hand side. Here, the second carbon atom, OH is on the left-hand side. This is D-mannose, and this is D-mannose. So therefore, they are called as C2 epimers. So at second carbon atom, the position of the OH groups are different. Hence, it is called as C2 epimers. If the OH groups are different, the first carbon atom, they're called as anomers. Other than that, it will be called as epimers. Name the chiral carbon atom, number the chiral carbon atom in cyclic beta D plus glucose. In cyclic beta D plus glucose. Now I will write the structure here itself so that it becomes easy. Now I'll erase this mannose structure. And I will write the cyclic structure of one of the form of glucose. Instead of writing CHO, we'll write C double bond O and H. Now we know that the fifth hydrogen atom of the OH group of the fifth carbon atom reacts with the oxygen atom of the first carbon atom. Therefore, the pi bond breaks, and now the first carbon atom and the fifth carbon atom are bonded through oxygen. Now they're asking number of chiral carbon atom. Now carbon bonded to 4 different groups is called as a chiral carbon atom. So this carbon is bonded hydrogen, OH, the whole group here, and the fourth bond is with oxygen. Similarly, this also is bonded H, OH. This is whole group, and this is a different chiral. This also is chiral, this also is chiral, and this also is chiral. So chiral carbon atom means carbon atom bonded to 4 different groups is called as chiral carbon atom. So in the ring structure also, glucose has 5 chiral carbon atom. Hence the answer for this question is option 1. The next question. Now, if you observe here, this is glucose. This is alpha D plus glucose, and this is beta D plus glucose. It is glucose, the ring structure there. The above process in which alpha and beta form, alpha and beta, they've written this A and B, alpha and beta form remain in equilibrium with cyclic form, and a change in optical rotation is observed, which is called as mutarotation. Mutarotation is the difference in specific rotation of plane polarized light due to the change in equilibrium between 2 anomers. What is mutarotation? It is a difference. It is a difference in specific rotation of the plane polarized light. PPL stands for plane polarized light due to the change in equilibrium. Due to the change in equilibrium between 2 anomers. 2 anomers in the solution. 2 anomers in the solution. Therefore, the answer for this is mutarotation. The next question, which of the following aldohexose gives the same osazone derivative? Aldohexose. Now, all are aldohexose. Which of the following aldohexose? What do you mean by aldohexose? Sugars containing 6 carbon atom and aldehyde group is called as aldohexose. So all contain 6 carbon, 1, 2, 3, 4, 5, and 6. All contain 6 carbon atoms and an aldehyde group. Therefore, all will be an example for aldohexose. But they're asking which of the following aldohexose give the same osazone derivative. The answer is option 2. The first and the third one will give the same number of, they'll give the same osazone derivative. The next question, osazone derivative we learned by the reaction of glucose with phenyl hydrazine. The next question is, how many moles of, there should be a bond here, how many moles of acetic anhydride is needed to react completely with tartaric acid, ribose, and glucose respectively? How many moles of acetic anhydride required or needed to react completely? Now, acetic anhydride, if I write an alcohol, OH here, if we write acetic anhydride, it is CH3, C double bond O, O, C double bond O, and CH3. This is acetic anhydride. Now, if this bonds break here, this hydrogen attacks oxygen and gets eliminated as a byproduct, acetic acid. In place of H, this carbon gets bonded. So we get RO, C double bond O, and CH3. Esters are formed. Now, since the tartaric acid has 2 OH group, the tartaric acid will require 2 moles of acetic anhydride. Now, if you observe this, ribose, it has 1, 2, 3, and 4 OH group, 4 acetic anhydride. Here 1, 2, 3, 4, and 5. Therefore, here 5 moles of acetic anhydride required. Therefore, the answer for this question is 2, 4, and 5. It is option 1. The next question, glucose on reduction with sodium in mercury or sodium amalgam is called as, and water gives. Glucose we write like the CHO, 1, 2, 3, 4, 5, and the 6th carbonate we can represent like this. You can write OH, H, OH, OH, all this is structure. Now this, when it's allowed to react with sodium, mercury, and water, it undergoes reduction. It undergoes reduction. When it undergoes reduction, what happens is this aldehyde will be converted to alcohol. Aldehyde will be converted to alcohol. So this is called as sorbitol. The molecule which is formed is called as sorbitol. Hence the answer is option 1. The next question, which of the following pairs give phenylosazone? Which of the following pair gives phenylosazone? It is direct question. Glucose and mannose gives phenylosazone. Hence option is 3. The next question, the configuration of chirality center in D-triose shown are, chirality in D-triose is shown as. Now, we can write an aldehyde group like this, CHO, and we have CHOH, and we have OH, H, and CH2, OH. Now, one more representation if we write, we have an aldehyde group here. We have COH, H, COH, H, and CH2OH. Now, this is D-triose. Triose means sugar containing 3 carbon atom and aldehyde. And this is L-triose. L-triose. This D-triose is represented otherwise by a minus sign, negative sign, triose. And this is represented by plus triose. Now, if we observe the rotation of this molecule, this is a chiral carbon atom. These two are chiral carbon atoms. Here, this rotates. This is priority 1, 2, and this will give the third one. So this part of the molecule rotates the plane polarized right to the right side. Therefore, this is called as, represented as are called as rectors in foreign language, in Latin. And here, this is OH1. This is sinister. It will rotate the plane polarized like anti-clockwise like this. This is the priority 1, 2, and 3 anti-clockwise. So this carbon rotates it in the left side. So it is called a sinister. Levo-rotated left side. Now the question, the configuration of the chirality center in D-trios is, now at the second position, we have sinister S, 3S. That is second carbon atom is 2S, sorry, 2S. And the third carbon atom is 3R, right side. Hence the answer for this question is option 3, 2S and 3R. Therefore, the answer for this question is 1 and 2, bromine water and furfural test. Bromine water test we would have learned. Furfural is a chemical test used to distinguish between glucose and fructose. The furfural test is similar to Molish test, but uses concentrated HCl instead of sulfuric acid, concentrated. And the solution is boiled. So glucose and fructose can be distinguished by bromine water test and furfural test. Therefore, it is option 2. Option 3, 1 and 2. The 26th question. 1 mole of glucose on respiration produces 36 molecules of ATP, 34 moles of ATP, 34 moles of ATP, 40 moles of ATP, 38 moles of ATP. The answer is option 4, 38 moles of ATPs. 1 mole of glucose gives 38 moles of ATPs. Now, the correct corresponding order of names of 4 aldoses with configuration given below. The correct order. The first one will be called as D-erythrose. The second one will be called as D-threose. The third, it's called as L-erythrose. And the fourth will be called as L-threose. Therefore, the answer for this question is option 4, D-erythrose, D-threose, L-erythrose, and L-threose. Answer is option 4. The correct statement in RNA and DNA respectively is sugar component in RNA is ribose and the sugar component in DNA is 2-deoxyribose. Correct? The sugar component in RNA is arabinose. It's wrong. And DNA is ribose. It's wrong. It is also wrong. It's also wrong. It's also wrong. It's also. Therefore, the correct statement is the sugar component in RNA is ribose and in DNA is 2-deoxyribose. The next question. D plus glucose reacts with hydroxylamine and yields an oxime. The structure of oxime would be. Now, glucose is this. I'm writing the structure, shortened structure. Now, hydroxylamine, it has a formula, NH2OH. Now, this undergoes a condensation reaction. So what happens is H2 and O gets eliminated as water. In place of O, this nitrogen gets bonded so that you get HC, double bond N, OH, the rest of the configuration remains the same. Therefore, the answer for this question is option 4, CH double bond NOH. You can see here it is CH double bond NOH. The rest remains same. Therefore, it is option 3 or 3, not 4. It is 3. It is 3 and 3. 4, because in glucose, the third carbon atom, the hydrogen is on the right hand side. Therefore, it is option 4. It is not 3. Option 4. So they've just confused here, second and third carbon atoms. So this is right. The third option is wrong. The next question. Which one of the following sets of monosaccharides form sucrose? Which one of the following sets of monosaccharide forms sucrose? One is alpha D glucopyranose, the other one is beta D fructofuranose. So glucose is made of sucrose is made of glucose unit and fructose unit. In more specific, alpha D glucopyranose and beta D fructofuranose. Therefore, the answer is 2, option 2. The difference between amylose and amylopectin is amylopectin have 1,4-alpha linkage and 1,6-alpha linkage. Therefore, the answer is option 1. It is a branch. Amylopectin is a branched chain. Amylose is it's a linear polymer. It is not a branched polymer. So therefore, the answer is option 1. A non-reducing sugar among the following is non-reducing sugar. It's a direct question. The answer is the third one, that's sucrose. The next question. Cellulose is a straight chain polysaccharide composed of only D-glucose units joined by alpha glycosidic linkage, D-glucose unit joined by beta glycosidic linkage, D-galactose units joined by alpha glycosidic linkage, D-galactose units joined by beta glycosidic linkage. The answer is option 2. Cellulose is a straight chain polysaccharide composed of only D-glucose units joined by beta glycosidic linkage. The incorrect statement about carbohydrate is maltose is a reducing sugar. True. Carbohydrate is stored in animal body as glycogen. That's also true. Amylopectin consists of 80 to 85% starch. Therefore, this is wrong. Amylose contains 15 to 20% starch. And the cellulose is composed of beta-D-glucose. This is also right. The incorrect statement is option. Which of the following carbohydrates is not oxidized by bromine and water? Which of the following carbohydrate is not oxidized by bromine and water? Which of the following is not oxidized by bromine and water? The answer for this is D-fructose. D-fructose is not, which of the carbohydrate is not oxidized by bromine and water. 36th question. Alpha D glucose is dissolved in water and kept for few hours. The major constituent present in the solution is. The major constituent present in the solution is alpha D glucose, the mixture of beta D glucose and open chain D glucose, a mixture of alpha D glucose and beta D glucose. So it will undergo mutarotation and it will have a mixture of alpha D and beta D glucose. Hence the answer for this question is option 4. The component that does not reduce Benedict solution. So which is not a reducing sugar. Sucrose is not a reducing sugar. Therefore, it will not reduce Benedict's solution. The next is amino acids and proteins. We'll finish with amino acids and proteins. During the process of digestion, proteins present in food materials are hydrolyzed into amino acids. We all know that proteins on hydrolysis gives alpha amino acids. Alpha amino acids. The two types of enzymes involved in the processes, proteins Enzyme A, polypeptides. Enzyme B, amino acids. So the two types of enzymes. So proteins with enzymes pepsin breaks down to polypeptides. And this polypeptide, in the presence of the enzyme trypsin, gives alpha amino acid. Hence, the answer is option 3. The next question, which of the following structures represent a peptide? It's a repeated question. Which of the following structure represents a peptide linkage? It is CONH, and this is CON. Therefore, it's option. It's a repeated question. We learned it in carbohydrates also. The helical structure of protein is established by dipeptide bonds, hydrogen bonds, ether bonds, peptide bonds. It is established by hydrogen bonds. Established by hydrogen bonds. The next one. An amine among the following that acts as a hormone is thyroxine, oxypurin, insulin, progesterone. The amine among the following, the amine. So all these are all these are hormones, no doubt. The amine among the following is thyroxine. Thyroxine is an enzyme which has a NH2 group. The next one, the hormone that contains iodine is insulin, testosterone, adrenaline, thyroxine. The hormone that contains iodine is also thyroxine. The sixth one, which type of amino acid is lysine? Which type of amino acid is lysine? So the structure of lysine is this. Since it has two NH2 group and one COH group, this amino acid lysine is basic in nature. Basic in nature. It's an example for basic amino acid. Hence the answer for this question is option 3. Excuse me. The structure of glycine is it's this. Just written on. The correct statement among the following is glycine is this. This is glycine. The zwitterionic form of glycine is HCH, COO minus, and NH3 plus. The H plus ion will be given to NH2. This is the structure of glycine. So the correct statement among the following is glycine as well as other amino acids are amphoteric. True. The acidic functional group in amino acid is alpha amino acid is NH3 plus acidic functional group. The basic functional group is COO minus. They have written as CO2 minus. That's also true. And all the statements above are correct. So the structure of glycine, the correct statement among the following is all the 3 statements are true. The next question. NH2CH double bond O. No, not this. This is NH2 CH CH3 C double bond O ENH CH2 COH. Identify the amino acid obtained by hydrolysis of the above. Identify the amino acid obtained by hydrolysis. When we hydrolyze this, we can write OH here and H here. So we get NH2 CH CH3 COH. This is alanine. And here you can see it's NH2 CH H COH. This is glycine. This is glycine. So this peptide, dipeptide, this is called as a dipeptide. When hydrolyzed, we get 2 amino acids. That's glycine and alanine. Hence, the answer is option 3, both 1 and 2. At isoelectric point, Concentration of cation is equal to concentration of anion. Net charge is 0. Maximum concentration of dipolar ion will be present, all the above. The isoelectric pH, concentration of cation is equal to anion, net charge is 0. Maximum concentration of dipolar ions which are present. Hence, the answer for this question is option 4, all the above. Which of the following amino acids has lowest isoelectric point? So lowest isoelectric point amongst this is given for aspartic acid. A protein that is a high is in a pH region below its isoelectric point will be positively charged, so it will migrate towards cathode. So a protein that is in a pH region between the below the isoelectric point will have positive charge, will migrate towards the cathode. So the isoelectric pH is represented as PL, calculated by the formula half into pK1 plus pK3. So this will be given as 1 by 1.8 plus 3.65. That will be equal to 2.77. The neutral form is dominant between 1.88 and 3.67. Therefore, the answer for this question is option 3. The next question. You can see here this is for the reference. PK and PL value. PK is the dissociating constant. PK calculated by the formula negative logarithm of K. PL can assign isoelectric point minuses. The next one. Which amino acid does not contain chiral center? It's a direct question. Only alpha amino acid which is not having a chiral center is glycine, which is not optically active. This is not optically active. Only amino acid is glycine. So we can write optically inactive, we can write. So glycine is optically inactive. The next question. Protein mainly contain carbon, hydrogen, oxygen, nitrogen, carbon and hydrogen, carbon, nitrogen, oxygen, nitrogen. So protein mainly contains carbon, hydrogen, oxygen, and nitrogen. The next question. Which statement about protein is wrong? Proteins occur in all living cells. Proteins invariably contain nitrogen, oxygen, carbon, hydrogen. Proteins are synthesized by plant kingdom only, which is the wrong statement. So therefore, the answer is option 3. Protein is also synthesized in the laboratory. That is for true. The main structural feature of protein is ester linkage, the ester linkage, peptide linkage, all these. So the main structural feature of protein is peptide. This printing mistake here. 2 options are given same. So the linkage in protein is a peptide linkage. The next question. Protein gives violet color with alkaline copper sulfate, forms a purple color with boiling with dilute in inhydrated solution. It's yellow color on boiling with nitric acid, all the above. The answer for this question is option 4, all the above. Protein gives violet color with alkaline copper sulfate, purple color with dilute in inhydrated solution, and gives yellow color with nitric acid concentrated. Keratin, a structural protein, is present in hair, skin, wool, all the above. So keratin is a structural protein present in hair, skin, wool, all the above. Answer is 4. Blood protein is albumin, hemoglobin, both 1 and 2, none of these. The blood protein is both albumin and hemoglobin. Hence, the answer for this question is option 3, 1 and 2. The destruction of the biological nature and activity of protein by heat or chemical agent is called as dehydration, denaturation, denitrogenation, deamination. It is denaturation. So therefore, the answer is option 2. Enzyme trypsin converts amino acid into protein, glucose into glycogen, starch to sugar, protein to amino acid. It's protein to amino acid. We learned the first question in proteins. The non-protein substances which certain enzymes require for activity is called as coenzymes. Non-protein substances which certain enzymes require for the activity is called as coenzymes. Antibodies are carbohydrates,